In this lecture, we will discuss the physiological process known as blood coagulation, which is also known, particularly in the clinical world, as hemostasis. There's no reading assignment for this particular lecture in our textbook, so you will simply have to rely upon the information provided in this lecture. Here are my learning objectives for this lecture, the things that I would expect you to be able to do after you've studied this material. Let's start with a patient. This particular patient has arrived at the emergency room with the following symptoms. They have a blinding headache on one side of their head, but oddly enough, it seems to be totally localized to the one side. They're also having great difficulty in forming their words and communicating with people, and they seem to be partially paralyzed on one side of their body. I'm sure many of you have heard of these kinds of symptoms before and can guess what's going on. But the question is, what's happened in this patient to cause these symptoms? And a further question is, what treatment might be administered to the patient after some kind of diagnostic scans have been run that might prevent severe complications of the event that's going on in the patient? We'll have discussed all of these things by the end of the lecture. So hemostasis is the maintenance of a closed circulatory system. Now this is distinctly different from the buffering of the blood serum, which is an open system, because part of the buffer is actually able to leave the system through interaction with the lungs. The blood, on the other hand, does not leave, and in fact we have a limited supply of blood cells and other things, such that if we develop a significant leak, it's life-threatening and repairs have to happen quite rapidly. This repair process involves both cells that originate in the bone marrow, these are called platelets, and also proteins, a wide array of proteins actually, which are mostly synthesized in the liver. And before we're done, we'll discuss the roles of both the platelets and the various proteins that are involved in the process. Hemostasis occurs in three phases. In the first phase, which is called procoagulation, what happens is that damage to the circulatory system, in other words, a blood vessel that's been damaged such that it's leaking and not maintaining or containing its materials, is recognized, and upon this recognition of damage, several things happen. Platelets are bound and they're activated and they aggregate, as we'll discuss, and to stabilize this plug that's formed by the aggregated platelets, a fibrous protein called fibrin is produced from a soluble precursor known as fibrinogen. The second phase is anticoagulation, in other words, turning off the procoagulation proteins to prevent excessive formation of this fibrous protein, because if you get too much, then you block off the blood vessel entirely, and that can have disastrous downstream consequences. Finally, the third phase of hemostasis, after a leak has been discovered and repairs have been initiated and are ongoing, the last step is something called fibrinolysis, and that is the actual systematic and orderly removal of the clot after it's no longer needed. So first, let's talk about platelets. Platelets are very small cells. They have, during the course of their development, eliminated their nucleus, so they can't make new proteins. They have all the proteins they're ever going to need, and a lot of other things that go along with the nucleus have also been discarded. So I said these are very small cells. In fact, essentially they're cell fragments made from a precursor called a megakaryocyte, but that's not really important enough for you to remember or memorize. And the platelets, of which there are a lot, some 100,000 per milliliter of blood, play three significant roles in the process of hemostasis. In the first role is binding. So a platelet moving along inside the blood vessel encounters a place where the endothelial cells have been removed, and these are the cells that line the surface of the blood vessel, exposing the subendothelial layer, a large portion of which consists of the protein collagen. The platelet binds to this collagen and interacts with another protein that's also located there, the membrane-bound form of a protein called von Willebrand factor. And through this activation, this interaction with von Willebrand factor and collagen as well, the platelet becomes activated. The activated platelet is a secretory cell. It secretes all kinds of things. It secretes chemicals to attract other platelets. It secretes chemicals to cause vasoconstriction, to cut down on the local blood flow, to make repair process more effective. It secretes growth factors to induce the growth of new cells across this hole in the blood vessel wall. It secretes proteins that are involved in the coagulation process called factors 5 and factor 13, and we'll discuss those at length later on as well. Now, after the secretory phase has gone on, the activated platelet then does something else. The chemokines, the chemoattractant molecules that are released, attract other platelets. And that brings us to the third role of the platelets. They aggregate together, they stick and form a clump that physically blocks the hole or the leak in the blood vessel wall. And this is facilitated in part by all these spiky protrusions that come up on the platelets after they've been activated. This process forms something called a soft clot, and if the platelets are functioning properly, it happens pretty rapidly. Unfortunately, this soft clot is not, this platelet plug can easily be wiped away, and is not strong enough to withstand the pressure of blood moving through the blood vessel. And that's where the proteins that are involved in the coagulation process come in. So to summarize, they bind to exposed subendothelial layers, basically the basement membrane that the endothelial cells grow upon, mostly to collagen. Another platelet glycoprotein in its membrane interacts with von Willebrand factor that's also present in that basement membrane, and this stimulates several signaling cascades that culminate in the release of chemokines, etc. One point that I didn't make on the previous slide that is important is that aspirin therapy, which you've all heard about with respect to heart disease, actually targets one of the key signaling molecules released by activated platelets. So this is where aspirin would play a role in preventing downstream damage from an inappropriate blood clot. The aggravation phase involves the platelets sticking together. And again, this plug, which will cover the hole in the blood vessel wall, isn't strong enough to stand up to full pressure when the blood flow has been reestablished. In other words, after the vasoconstrictor molecules secreted by the platelets have diffused away. Stabilization of the platelet plug is where the proteins involved in procoagulation come into play. And the protein's role consists of two interconnected cascades of protease enzymes, in which one activates the next and activates the next, both of which lead to the activation of a single protein, a protease called factor 10. And that's going to be generally true. The proteins involved in coagulation are generally named factors and assigned a Roman numeral. So this protein here would be called factor 12. When it becomes activated, it would become called factor 12A. So we have two interconnecting pathways, the intrinsic pathway and the extrinsic pathway, which are each activated through separate signals, separate proteins recognizing separate things that suggest that there's a hole in the blood vessel. And they lead to the activation of factor 10, which starts a final common pathway, which will lead ultimately to the activation of a protein called thrombin that has multiple roles, including converting the precursor for the main protein that's going to form the clot fiber, a protein called fibrin, into the active form. Both the extrinsic and intrinsic pathways lead to the activation of factor 10. And factor 10 then, on the next slide, not shown here, is going to activate factor 2, which is prothrombin. Let's look first at the intrinsic pathway. And this is initiated by the formation of a large complex of proteins, including something called high molecular weight kininogen, a protein called prekallikrein, and factor 12. And this complex formation ultimately leads to the activation of factor 12 to factor 12A, which is an active protease. Factor 12A then will activate another protein called factor 11 to factor 11A. This becomes the serine protease. This activates a protein called factor 9 into factor 9A, and factor 9A activates factor 10 to form factor 10A. So you notice that we have multiple steps, and just like when we had a kinase cascade for signaling, this provides amplification, because if each activated protease only acts a few times, then we get a geometric activation, so we get lots of activated factor 10. On the other side, on the intrinsic pathway, this involves a circulating protein called factor 7 binding to a membrane-bound protein called tissue factor, and tissue factor is also known as factor 3. This binding interaction converts factor 7 into an active protease, and it then directly activates factor 10 to make factor 10A. And remember, this is the ultimate goal of both arms of this pathway. The intrinsic and extrinsic arms are both centered around the activation of factor 10. But the two arms are not completely independent, because factor 12A has a second substrate besides factor 11, and that is factor 7, activating some factor 7, and factor 7A has another substrate as well, which is factor 9, converting it to factor 9A. So they're interdependent, there's crosstalk, and you can't get by without both sides, because due to factor 7 having some activation role in factor 9A, you have to have it. And even though you can get by without factor 12 or factor 11, you can't get by without factor 7. And we know that that means that we have to have factor 9 activation through factor 7. So here's the same thing in words. I won't spend the time reading this to you, but you can certainly look through it yourself. And the key point, two key points. We're going to activate factor 10 to factor 10A, and the extrinsic and intrinsic arms of the coagulation process are not independent of each other. That leads then to the final common pathway in which ultimately fibrinogen, a soluble precursor protein, is going to be converted into a fibrous protein called fibrin that will stabilize the platelet plug and seal off the leak in the blood vessel so that repairs can occur. Here's how the common pathway works. Factor 10A is activating another protein known as factor 2, or also known as prothrombin, and factor 2A is called thrombin. Thrombin has several important activities. The first is that it converts fibrinogen into fibrin, and we'll talk about that on the next slide. Remember, that's the ultimate goal, to form the fibrous protein to stabilize the platelet plug. Thrombin also has a feed-forward activation loop. It activates some factor 12, and you remember that's up at the top of the intrinsic pathway. It also activates two other factors that we haven't talked about yet, factors 5 and factor 8. And those lead, respectively, to thrombin activation and factor 10 activation. So through these activities, activating factors 5 and 8, thrombin greatly increases thrombin activation. Finally, it also activates another protein yet, which is called factor 13, which is going to carry out cross-linking of the fibrin molecules, making a very stable, secure meshwork. And you should be thinking at this point of collagen, where cross-linking was the final step, making a very strong fibrous protein for its many purposes in the body. So here's how factor 8 and factor 5 work. Factor 8A, after it's been activated by thrombin, has two binding sites. One of those is for factor 9A, and you'll recall that's a protease that activates factor 10, and the other binding site is for factor 10. So this factor 8A holds these two together, kind of like a scaffold, and enables factor 9A to activate factor 10 much more rapidly, leading to a great increase in factor 10 activation, or in other words, more factor 10A. And remember, 10A is the protein that's going to actually activate prothrombin. Factor 5A does largely the same thing, except it binds factor 10A and prothrombin. Again, holding them together, decreasing the amount of time they have to spend diffusing around looking for each other, and greatly accelerating thrombin activation. This is one of the few places in biology and biochemistry where you actually see a feed-forward loop for activation, because once you have this going on, it's at least theoretically possible to go far too far and get greatly excessive activation of the protein. And since these are serine proteases, you could lead to extensive tissue damage if this went on too much, not to mention the problem of forming too much fibrin meshwork and gumming up the whole blood vessel works. We'll get back to the negative, or the turn-off mechanism, in a little bit. First though, let's talk about fibrinogen. Fibrinogen is a soluble protein, and it consists of multiple polypeptides interacting together, in fact 3 of them, forming a structure that's actually remarkably similar to collagen. The main difference being that the fibrinogen has 3 globular domains, one on either end and one in the middle, and the proteins are actually held together covalently by way of cross-links of the gamma chains. Fibrinogen itself is soluble, and the main reason that it's soluble is that on the N-terminal tails of these 3 polypeptides, really 6 polypeptides that are coming together, there are lots of negative charges. There are lots of aspartic acid residues, there are a lot of glutamic acid residues, and also there's a third residue, tyrosine residues that have been post-translationally modified to add sulfate, which is going to carry negative charge as well. So these N-terminal peptides, which wind up in the middle of this fibrinogen, have a great deal of negative charge, and you'll recall that like charges repel each other. So the fibrinogens repel each other electrostatically. What thrombin does is removes these N-terminal peptides through a proteolytic cleavage, converting the protein into something that no longer repels each other, and just like collagen, it spontaneously aggregates through the alignment of its triple helical regions, shown here schematically up above, interspersed with the globular regions. So it forms a very rapid self-aggregating meshwork. This protein, while strong, still isn't strong enough. And just like collagen had to be, to make it a truly strong and stable fiber, you have to cross-link the various fibrin molecules to each other. We'll talk about that in a moment too. The cross-linking of fibrin is not exactly the same as the cross-linking of collagen. You'll recall that that involved the oxidation of lysine residues and then spontaneous cross-linking with other lysine residues. In this case, we use a protein called a transpeptidase that is known as factor 13A that catalyzes a conversion of the soft clot into a stable hard clot that can stand up to the pressures of an active blood vessel. So what this protein does, factor 13A, is takes lysine residues on one fibrin molecule and glutamine residues on another fibrin molecule and interacts so that the amine side chain of the lysine replaces the amino portion of the side chain of the glutamine, forming a peptide bond between the two side chains, an amide crosslink. And we call this an isopeptide bond because it's involved in the side chains rather than in the backbone. So the key thing to remember is that this reaction is carried out by factor 13A, and it just involves cross-linking between glutamine and lysine residues. And this leaves us with a hard clot. And now, once we form this hard clot, then the platelet plug isn't going anywhere, and repairs to the blood vessel can be made. Since these involve growing new cells, they're going to take some time. All of this effort, all this conversion of fibrinogen into fibrin happens on the membranes, or at the membranes, of the platelet cells themselves, right exactly where you want it. And this is accomplished through requiring a number of the clotting factors, specifically 2, 7, 9, and 10, or thrombin. This is the activation protein, the top of the extrinsic arm. Factors 9 and 10, you remember, the bottom of the two arms. And these are only active when calcium is bound. Calcium, which was at one time called factor 4 because we didn't know what it was eventually figured out to be calcium ions, localize all these proteins to the platelet membrane by binding them through a post-translationally modified amino acid called gamma-carboxyglutamate, formed by adding an additional carboxyl group to the glutamate side chain. And what this does is forms an arrangement of oxygen atoms that is just the perfect size and shape for binding calcium. The calcium atoms are bound on the other side to phospholipids in the platelet cell membrane, localizing these factors then where? To the outer membrane, outer face of the plasma membrane of the platelets. So this carboxylation of the glutamate is critical for forming active coagulation factors. So let's talk about that process next. The formation of these gamma-carboxyglutamates, which are abbreviated GLA, requires another molecule called vitamin K. And in the reaction, carbon dioxide, or actually probably bicarbonate, is added by way of an enzyme that requires, among other things, vitamin K. And vitamin K, the quinone, in its reduced form, provides reducing equivalents to allow the addition of the bicarbonate to form a carboxyl group on the glutamate residue. In order for this to keep occurring, then the product, the vitamin K product of the reaction, has to be converted back into the active form through a couple of reductase enzymes. And the inhibition of these reductase enzymes by drugs like warfarin, which I'm sure you've heard of, one of the more commonly prescribed medications for people who have excessive clotting problems, then cuts down, doesn't eliminate, but cuts down on the fraction of all these proteins, factors 2, 7, 9, and 10, that have appropriately carboxylated glutamates. And since they're only active in the presence of calcium, and if they don't have the gamma-carboxyglutamate, then they're unable to bind calcium and they can't be activated. You don't want to eliminate it on all of the proteins, just some of them. So competitive inhibitors, like warfarin, are a very effective way to cut back on excessive clotting. What I want you to remember from this is simply that these drugs target vitamin K, and vitamin K is essential for the carboxylation of these glutamate residues, providing calcium binding activities. So to summarize, procoagulation consists of two separate but interdependent pathways leading to factor 10 activation. Factor 10 then activates thrombin, which then does several things. It accelerates its own activation, and it converts fibrinogen into fibrin, and it also activates the cross-linking protein. And both these cascades are activated through the exposure of proteins that are seen in the subendothelial layer, collagen or tissue factor. Now let's talk about the second phase of hemostasis, and this is called anticoagulation. Just like we've talked about before, every process that can be turned on through some regulatory pathway, coagulation has an off switch. And if you didn't have this, you'd build up excessive fibrin meshworks that would eventually block off blood flow completely, ultimately leading to death of the patient. Thrombin binding to its receptor, another protein yet, which I haven't talked about yet, actually turns on anticoagulation by stimulating the release of a protein called protein C. This protein C will form a complex with the circulating protein called protein S, converting protein C into an active protease. And the protease activity of protein C has two targets, factors 5A and 8A. So what does this do? Remember that factor 8A is involved in accelerating factor 10 activation, and factor 5A is involved in accelerating thrombin activation. So by degrading these proteins, then we're turning off the activation of thrombin. We're shutting down that feed-forward activation loop. But we still have to deal with the active proteases that we have. We're not making any more, but we still have a bunch of thrombin. We have a bunch of factor 10. We have a fair bit of factor 9 that are active. We have to deal with those as well so that we really can turn off the cascade. And this happens by the proteins irreversibly interacting with small proteins known as a small protein known as antithrombin 3. And antithrombin 3 is a protein in the class that's called serine protease inhibitors. And what it does is it binds to these activated proteases and goes through part of the reaction cycle with them, covalently modifying the enzymes and leaving them dead. Here's how it works. And again, there's going to be some reminiscence between this and the actual roles of factor 5 and 8. The activated proteases, factors 10A, 9A, and thrombin, have positively charged surfaces that allow them to interact with a molecule we also heard about recently, heparin. Here's the repeating dimer of the heparin, which is one of those glycosaminoglycans we talked about in the previous lecture. And the thing to notice is here we have a carboxylic acid, which at physiological pH is going to be negatively charged. We have a sulfate, we have another sulfate, we have yet another sulfate. So this glycosaminoglycan has 50 odd sugar units with 2 negative charges each. In other words, it's massively negatively charged. And the positively charged surfaces of these active proteases cause them to bind to the heparin. Antithrombin 3 also has a positively charged surface, so it interacts in conveniently a complementary way. In other words, the positive charges are just in the right place to interact with the negative charges in the heparin, so that all 3 molecules are held together, and the heparin serves as a scaffold to hold the antithrombin 3 and the active proteases together, allowing the antithrombin to turn off the activated proteases. For this reason, heparin is frequently used as an anticoagulant molecule, particularly if some procedure is going on that requires coagulation to be decreased, or particularly in patients who suffer from excessive coagulation problems. Finally, the last part of the process is called fibrinolysis. Again, it's great to be able to form a clot, physically plug a hole in the wall of the blood vessel so that repairs can happen. Once those repairs have happened, now we have to safely get rid of it because it can stick to things, and we don't want to block blood flow once repairs have happened. So that's fibrinolysis. Fibrinolysis is carried out by a protein called plasmin. This is a protease, has one substrate, and that is fibrin. The precursor for plasmin, the inactive precursor, like all these proteins, which were synthesized as inactive precursors, has a high affinity for fibrin. So it binds to the fibrin as it's forming before it's cross-linked and gets physically incorporated into the fibrin meshwork. And while it's there, as the inactive form, it doesn't do anything. It just sits there waiting for activation. It's activated by a protein released by nearby endothelial cells called tissue plasminogen activator. So tissue plasminogen activator, sometimes called TPA, is secreted from the endothelial cells when protein C, protein S complex, which you'll remember was one of the key players in turning off procoagulation, binds to a receptor. The tissue plasminogen activator then diffuses to the clot where it cleaves the fibrin protein, where it cleaves the plasminogen into plasmin, and the plasmin then can degrade the fibrin meshwork into small pieces, allowing its safe removal and degradation. So here's how the regulation of anticoagulation and fibrinolysis go, this time in words. It's a relatively elegant system in which each phase of hemostasis is initiated before the previous phase is ended. So procoagulation results in the activation of thrombin. Thrombin does all kinds of things, including activating fibrinogen to make fibrin. When enough thrombin builds up, it's able to diffuse away from the platelet membrane, and bind to its receptor, that protein called thrombomodulin that we talked about briefly, stimulating the release of protein C. So in this way we have a timing loop that allows procoagulation only to be active for a particular time, and then it's shut off, and it's turned off by a key protein in the procoagulation, which is to say thrombin. Likewise, fibrinolysis is turned on before anticoagulation has even been turned off. And this occurs when we get enough protein C, protein S complex to accumulate for it to diffuse away to bind to a receptor on endothelial cells and then stimulate tissue plasminogen activator release. Finally, fibrinolysis is going to be turned off by antiproteases, antiplasmin and plasminogen activator inhibitor. And these proteases then will bind to their respective targets, plasmin and tissue plasminogen activator, and irreversibly inhibit them, turning the process off. So back to our patient that we started with. This particular patient was having a problem known as a stroke. The stroke is what happens when blood flow in the brain, some region of the brain, is restricted, preventing oxygen and other nutrients to reach the neurons that need a lot of oxygen and nutrients that are behind where the blockade happens. About half of all strokes happen when a blood clot forms in the brain and blocks that artery, causing damage to the cells behind them. Once a diagnostic scan like an MRI or something like that has been done to ensure that the stroke is not due to a hemorrhage, in other words excessive blood leaking, then the patients that are having a stroke because of a blood clot in their brain are treated nowadays with tissue plasminogen activator, which then does what? It activates the plasminogen in the blood clot and allows the degradation of the fibrin meshwork, allowing reestablishment of blood flow and preventing the death of the neurons. And in most cases this treatment is very effective and prevents all the things that used to happen when you had a stroke. A lot of people who had a stroke would wind up paralyzed on one side of their body permanently. These days, provided they get to the hospital fast enough, to the emergency room, they're diagnosed as having a stroke rapidly enough, and tissue plasminogen activator is administered, then in most cases there are very few long-term consequences. And that is what I want you to know about hemostasis.